Hello everyone, welcome to Ocean, and this is me Sheethal with you, and today we are here with our new video, again back with the cell series, and the topic is about Golgi complex, which is an important cell organelle. And we will be discussing more about Golgi complex in this video. And before getting into the video, if anyone of you are new to my channel, please go and subscribe to my channel and press the bell icon so that you can get the notifications whenever I upload a video. So, simply talking about Golgi complex, it has a lot of variety names, right? Golgi body, Golgi apparatus, everything is right. And we can call it generally as a Golgi complex. Okay, it was discovered by Camillo Golgi and the name goes after her. It is seen within the cytoplasm of both plant and animal cells and is composed of stacks of membrane-bound structures which is called as cisternae. Individual stalk is called as dictyosome. And mammalian cells have about 40 to 100 stacks. Each stack is in between 4 and 8 cisternae. Proteins have as many as 60 stacks. And cisternae comprises flat membrane-enclosed discs that includes special Golgi enzymes to modify or help the cargo proteins. And cisternae also carry structural proteins which are important for maintaining the flat membranes. Usually, this Golgi complex is found near the rough endoplasmic reticulum, and the cisternae stack is in 4 regions. Is seen in 4 regions, that is cis-Golgi, endo-Golgi, medial Golgi, and trans-Golgi. Each contain different enzymes controlling variety of functions. Cis-Golgi is found near to the endoplasmic reticulum, and Golgi apparatus receive cargo vesicles first here containing newly synthesized proteins. They fuse with the Golgi membrane and empty their contents in the lumen, and inside lumen molecules are modified. When the cargo proteins pass through the endo-Golgi and medial Golgi, most of the functions of Golgi apparatus occur. Eventually, it ends up in the trans-Golgi, located at the opposite side end from the cis-Golgi. Here, finished products are packed for transportation by placing them into one of the at least 3 different vesicles depending upon the molecular marker they carry. And this is how the transfer of materials through these 4 types of Golgis happen. And now let us understand what are vesicles. There are 3 major types of vesicles and we will be discussing about them in detail. So let's understand the different types of vesicle. The first type is exocytotic vesicle, which is also called as continuous vesicle. And it contains proteins destined for extracellular release. After packaging, they bud off and immediately move towards the plasma membrane and fuse and release contents into the extracellular space. And this process is called as constitutive secretion. And an example for this type of constitutive secretion is the antibody released by the activated plasma B cells. And the second type of vesicle is secretory vesicle, which is also called as a regulated vesicle. And it contains proteins destined for extracellular release. After packaging, they bud off and stored in the cell until a signal is given for its release. After receiving appropriate signal, they move towards the membrane and fuse to the released contents. And that is called as a regulated secretion. An example of this regulated secretion is a neurotransmitter release from the neurons. And the third and the last type of vesicle is lysosomal vesicle, and they contain proteins destined for lysosome or an organelle of degradation containing many acid hydrolases. Proteins include both digestive enzymes and membrane proteins, and vesicle first fuse with the late endosome. Contents are then transferred to lysosome through unknown mechanism. And one of the major process here happening is the digestive proteases destined for the lysosome. Altogether, Golgi body serves as a processing plant for the products manufactured by the endoplasmic reticulum. And now let us understand the functions of this Golgi complex. First of all, it helps in phosphorylation. Phosphorylation means it is the addition of phosphates. And also it helps in glycosylation, and that is the addition of carbohydrates. It helps in the production of polysaccharides and glycosaminoglycans, which is used in the manufacture of carbohydrates. It helps in synthesizing the proteoglycans and help in the breakdown of cargo proteins into small active fragments. An addition of sulfate group to some proteins through the help of sulfotransferases is also mediated by this Golgi body. Formation of lysosomes for the transportation of lipid to other parts of cells, and formation of coproteins by polymerization of glycosaminoglycans is also mediated by this Golgi bodies itself. BCL2 gene present in the Golgi body helps in the prevention of apoptosis, which is a programmed cell death. And these are all the functions that it employs a lot of functions which are like a huge diffused processes that occur all over the body, and hence it has a core impact on the body functions and all that. And before that we learned about the different apparatuses that we see in the Golgi complex, and also we understood about the whole structure, otherwise what we can see through it, morphological structure of Golgi complex and all like that. I hope you find this video helpful, and you can get I hope that you get a concept, a simple concept about the Golgi complex. If you find this video helpful, please do like, share and subscribe. And also, if you have any suggestions or if you have any kind of doubts, please let me know through my comment section, and please do share this video with everyone who is interested to learn biology. Thank you very much for watching, and we will be back with the next important and an exciting topic.